Ready. Yeah. Okay, so hello everyone. Thanks for joining us for today's seminar. It is my pleasure to introduce you to Dr. Sharik. He is the research professor and emeritus dean in Michigan Technological University. He is a nationally recognized leader in natural resources education and forest ecology, with over hundreds of publications and decades of service across different institutions, including Utah State University, University of Michigan Biological Station, and Virginia Tech. He is also the co-founder of major professional gatherings, such as the biennial conferences on university education in natural resources and the North America Forest Ecology workshops. And even in retirement, he still contributes significantly and remains active in advancing natural resources education and volunteers in several restoration projects. So please join me in welcoming Dr. Sharik.
Well, good afternoon on a Friday afternoon when people are clearly doing something other than they're doing right now. The first disclaimer is that I should mention that I have a research professor position, and I do, but I'm technically retired, a Emeritus faculty member. But I use that, Michigan Tech allows me to keep that that title so that when I apply for grants, people don't think I'm entirely over the hill. So it can be useful even though I'm not being paid for what I'm doing. So our institution is very good about it. Before starting into the formal presentation here, I'm painting this. I'm really want to focus on students, graduate students, particularly those who are going to be going out into the workforce and are going to be spending a career at this sort of stuff. In terms of, if when I go through this presentation, to think about this from the standpoint of your long-term career goals and how you're going to function in a job somewhere in the general area of conservation. Let's just say conservation-related jobs. So I took a few notes on this. The first thing is that the recovery of and restoration of long-lived species, such as tree species, requires that you do long-term investigations. And so the first question is, how do you maintain that over a period of time? We've been involved in this for a half century now. How do you engage other people, younger people, in the process so that you can continue and move those forward for time immemorial if you need to do that? Secondly, what is the right mix of basic and applied research that is appropriate to apply to these conservation efforts? Okay, and you'll find that when you, for example, in many, the common thing, if you're working for an environmental consulting agency, there simply isn't the funding to do research. And so you just get away from the research together. You do what you do is you try things. If they work, you retain them. If you don't, you try something else. So it's almost like intergenerational local knowledge, which is good, but it's not documented and made accessible then to the public later. So how do you still retain some of that basic and applied research that's needed to answer the applied questions that you want to answer? So that's the second point. The third point has to do with this mix of the biological and physical sciences. The biophysical sciences and the social sciences. And it's been my experience that in working with a lot of different people and students who go through programs that are biophysical or ecological in nature with little or no human dimensions of social sciences component to it. They get out into the workforce, and guess what? The problems on the human dimension side overwhelm the biological and ecological stuff. And so the old adage that the biology or the ecology is easy, it's the people that are the challenge. And it really is in so many sorts of conservation efforts. And the fourth thing is that despite what we may think as human beings, nature really is in control. And so when the challenges of setting up long-term sorts of investigations, and you put in for funding from an agency, and you're going to treat 4 areas in each 4 successive years, and then something happens in terms of the weather, insect infestations, and it blows a hole in the whole thing. And so how do you adjust? You got to remain flexible enough, and hopefully the organizations that fund you will remain, to allow you to go to plan B, which you necessarily have to do. So nature is in control ultimately, and we are not in control. And so you have to adjust that. Okay, so with that background, I want to get started here. I've been given an hour. I can take three hours. We're talking about a half-century worth of effort with the recovery of this federally endangered tree species in Virginia around these birds, Betula, over. So I'll move through, and I think I'll at least get through the most important elements related to the recovery plan, and then to the extent that I have time, I'll cover the others a little bit less so. The other thing is that, I think you'll run across this. There are a couple of ways of giving these sorts of presentations. One is that you just have material which is more of an outline sort of nature. And if you're there and you experience the presentation, then you learn something and you go away and you have something back. But if you're not in the presentation and you're looking at a sketchy outline afterwards, you can't figure out what's going on. I've taken the former approach so that you'll see that these slides are really pretty quote unquote dense. It's almost like looking at a publication. So if I just shared this with somebody who had never seen this before, they could make sense of the presentation. Okay, so in other words, you wouldn't have to take a note here. You can go back to this presentation and hear it here. The disadvantage is that it seems so much more scripted than it does if you're doing it off the cuff sort of thing. So that's the caveat in all of this. So let's get started. These are acknowledgements of the people who have helped since 20 and 21 on this project. No projects like this can be done in isolation. It requires a huge number of people. When we established what is de facto the recovery team over in the 1970s, late 1970s, we had as many as 23 people that served on the recovery teams for this effort. And mainly the organizations, the U.S. Forest Service, U.S. Fish and Wildlife Service, which of course has the responsibility for administering the Endangered Species Act, and the Virginia Department of Agriculture and Consumer Services, which has a responsibility for managing the Virginia Endangered Insect and Plant Act in the state of Virginia. So all of those 3 are major players, plus a number of other people. Bob Ford, who was a graduate student of mine when I was at Virginia Tech, is now retired, lives in Asheville, and he's a major player in this. And there are others, and I just wanted to mention, go to the next slide here. Suresh Speedy, who was a former PhD student of Mike's, is now at Norfolk State University, and he's involved in this project. We're just now in the process of submitting a proposal to the National Fish and Wildlife Foundation for additional work, and he's going to be the PI on one component of this. So he's a great person. He was well educated. He's a good person, and so I feel very good about involving him in this endeavor. So a little bit, if somebody wanted, I put in here this reference because I was asked by Virginia Forest to write this up, the history of all of this, and so if you wanted something that just gives that overview in a more common way, then this is a good reference to turn to do that. And then now what I want to do is move to a summary of the milestones over this half century. And I have 2 summary slides in here. So I'm just going to, because of the time, I'm just going to summarize some really key points historically over the last 50 years. And behind that are a bunch of other slides. And if you want to look in more detail at all of this, and especially the human dimension of what has happened over this last 50 years, then you can look at those in more detail. But This Virginia wildleaf birch was first discovered in 1914 by a person by the name of W. Ash. He worked a lot in the North Carolina area and Virginia area and so forth, along the banks of Dickey Creek in Smith County. Smith County is shown here as a county in Southwest Virginia, which is where this location is. And at 2,800 feet elevation. And then it was considered, people looked for it again, they couldn't find it. So it was deemed to be lost or extinct. But then in 1975, Doug Ogle, who was on the faculty at Virginia Highlands Community College, went back and started looking at adjacent watersheds. And lo and behold, he found it in an adjacent watershed, Crescent Creek watershed. And so at about the same elevation. So in all likelihood, what happened was that when Ash annotated his specimens, he actually thought that he was on Dickey Creek and he probably was on Crescent Creek. So everybody was looking in the wrong place for this thing. So it was rediscovered in 1975, which is the year I went on to the faculty, joined the faculty at Michigan Tech in 1975, and had done my PhD work on the ecological genetics of dark bark birches in the Appalachians, as luck would have it. So I was predisposed to getting involved in this. Anyhow, in 1977, we created this protection management research coordinating committee. Was formed, and this is a de facto. They didn't want to use the time the term recovery team, so we used this term, and that was in 1977. And then it's a year later in '78. It was officially listed as Endangered Species Act of 1973. It was the first tree species to be listed in that way. And then in '79, following year, it was listed as endangered at the state level under that Virginia Endangered Plant Insect Act. And interesting enough, it's coded into the act, so it's very hard to take it out. So even though we now are at the threat level federally, they still have it designated as endangered because they have to change the nature of the act itself. And because it occurs only in one location, they're saying it probably should always be considered endangered because it's only in one place. And if we lose it there, well, it's gone. So that was the rationale for that. In 1982, the recovery plan was approved under the Endangered Species Act, and it was the first plant that was for which there was a recovery plan under the Endangered Species Act. And so it was fun putting that together. I was using those recovery plans from animals to sort of try to figure out how I was going to put this together. And then jump ahead to 1994, and that's when the species then, as a result of our 2 decades or more of work, that it was downlisted from endangered to threatened over that period of time. When we established 20, what they want to use as a terminology, additional populations that were in the same watershed, in that same watershed, we now refer to them as progeny populations because they're progeny from open pollinated trees in the original population. And then 1923, now jump another space of time here, the first implementation of regeneration treatments in those progeny populations. And this is really critical of the whole story because establishing that natural regeneration in the progeny populations is fundamentally necessary to delist the species. So until you get there into this next generation, then it was not according to the recovery plan. It would not be delisted. And then so 23, we implemented these treatments in one of these 20 populations, and in 2024 we were successful in doing this. I'll show you how we did that. So that's the short story of all of this chronological stuff. And here I'm just going to skip through these. So then this recovery plan, as shown here in the folder of this plan, I just wanted to show you the overall goal that is shown here to increase the number of roundleaf birches in the wild to a point where it could be delisted. And in working with geneticists at the time, we came up with this estimate of 1,000 individual trees in each of 10 populations. And we later changed that to include 500 to 1,000 trees in each of 10 populations that are greater than 2 meters in height. In other words, we know that we're being successful. They're going to thin themselves out, but that we are at a level where that area that we're going to treat is going to be fully occupied by adult individuals in a reproductive stage. So I'm gonna take some water here. So if you look at that recovery, there are 6 major actions that are required under the recovery plan. And this is the basis for what I'm going to talk about in moving forward. And you see that at the time, we were required to put priorities on those, right? And so you see as the first priority, the maintenance and expansion of the natural population. And then right behind that is this establishment of these self-sustaining additional populations that I've mentioned wild. And then from there, continued searches for other populations are priority 2. And I can tell you the short story is that we've done exhaustive searches in other places, and we've never found it. So the probability of this vegetable ever occurring anywhere else is really, really small. Although we didn't have at the time, and you still don't have the techniques to look for those genes in the population. We're getting there. And then what are the biosystematic relationships? How is Betula uber related to other members of the dark bark birch group, especially sweet birch in particular, sweet birch Betula and Betula yellow birch Betula alliginius, all 3 of which co-occur in that environment. The retention of germplasm through cultivation, priority 3, and then I did the systematic really, and then finally implementation of education programs. So the areas that we're focusing on now was priority 1 and 2, and then 2 and 3. And the proposal that we just submitted covers those first. So it just covers those first areas. Okay, I'm going to go again to. Okay, so here's the first, the maintenance and expansion of the natural population. That's the first of those 6 major areas that we're dealing with. And so, and it's a distinct group of birches. You know anything about birches, but this group has really strong oil wintergreen in the inner bark and the foliage, which makes it unique in conjunction with direct female catkins at maturity. I'll be showing you some of those. These are short-lived, early successional species in timber regions, okay? Confined to the eastern US, and 2/3 species, depending on at what level you classify Betula over. It's always interesting with respect to the Endangered Species Act and what is possible and is not possible. What I can tell you that over that 50 years, taxonomists have classified this thing at the level of species, subspecies, variety, and form. Okay, all 4 of those, and it keeps dancing around. And and then the fact that where do these things occur? Typically cool, moist, montane type of environments, like in coves, especially in bordering small streams in the southern Appalachians. But also, this is a really important part of the story, as it turns out, Wilterno, also on rocky outcrops. And the microhabitat has been created by the rocky outcrops. And then, but the group is also, in terms of yellow birch, it's more widespread in the north, it gets up into New England and over into the lake states. And the key here is that requiring, because it's an early successional disturbance of related species, it really requires a disturbance to regenerate. You've got to have a paucity of overhead cover and no litter. You'll see that these are really, really small seeds, and the regeneration niche is very narrow. And it's like so many species, the bottleneck is in the regeneration niche, in that regeneration phase where it really occurs. And And then you look at it in terms of its tolerance to understory conditions. It's moderately intolerant of closed forest canopies. And as you'll see, of the individuals in our progeny populations that die, they're relegated to the understory. Once they get to the understory, they're toast, so to speak. If they can't, if they're not capturing light in the canopy to some degree, then you've got a real problem. So a little bit about the life history of these things, just so you understand that. To begin with, birches are monoecious. They have male and female flowers on the same individuals, and they occur in these clusters called catkins. These inflorescences are catkins. And here, this is in the springtime, and here is a female catkin in one bud here, and this is a male catkin here, which is expanded now and is ready to throw out pollen, right, in the spring. These things are wind pollinated, so that everything has to happen before leaf out, and it does early in the spring. And if you look at the flowers up close, here are the male flowers, no petals or sepals, they're just a bunch of stamens there, right? It's pollen sacs in the males. And then here are the females, shown like they're clustered in threes with their long styles to capture that incoming pollen. And you just want everything out of the way. You're dumping a lot of pollen out there in the wind. You don't want anything in the way, you just want it to affect fertilization. These things have strong barriers to selfing. And so that even though you have males and females on the same thing, the timing is different, and there are other barriers that well occur that preclude selfing. It's only about 1% of the individuals that ultimately produce the seed that would occur from pollen fertilizing the eggs of that same individual, which is good if you're rare and you need something out there, but otherwise it's discriminated against. So, and these things then, if you look in detail at this inflorescence and a side branch of it, that inflorescence, these are the females now, like we're looking up here. You tear that thing apart, it's a cymose inflorescence, and the side branches is shown here called cynules, a subdivision of that. And you'll notice that there are 3 flowers there, just like we were looking before, and then each one is subtended by a pair of bracts. And the evolutionary interpretation of the bracts that are dead is that they were eliminated, and 3 of these bracts were retained and coalesced until they formed one single bract here, 3-lobe bract with a flower and ultimately a fruit at the base of those 3 lobes. And you can see already that the wings are developing on those ovaries because they're going to be small winged nutlets, or Semeris as they call them, or wind dispersed. They're only 2 to 3 millimeters in length and width. So we're talking about a huge, really very, very small fruits, which are dumped out, by the way, I'll show you some figure, by the thousands from even a given tree. And yet there's no reproduction over 40 years, even though we're dumping out millions of seeds, and we know that a reasonable percentage of them are viable. So it gets back to this thing of the regeneration niche. You can put those things out there until forever. And if you don't have a proper regeneration niche, you're not going to get it. So now here are the females now at maturity in the fall. So these are after pollination, effective pollination. You've gone through the growing season, and now in the fall, these catkins are mature, usually starting in November, and they're going to disintegrate. So all of the bracts and all of the nutlets will come apart, and they'll be wind dispersed. And here, here's one hole, and I tore it apart. And over here are the bracts, the 3-lobe bracts, and then over here are the Semeris, the seedlets. And then just this past November, actually starting early, this is just a shot that I took on the snow. And you can see those bracts scattered around, along with here's a bract, right? And then here's the Semera, that little winged Semera. So they can be dispersed across snow too, so they're not only dispersed by wind from the trees directly, but if they're on across the snow, then they'll move across the snow as well. Seed germination and seedling emergence occurs in early spring under cool, moist conditions, and then you get rapid growth in the near absence of overhead cover, and as I say, in the absence of litter too. Otherwise, if they land on top of litter and it dries out, the radicals will emerge and they just shrivel up and gone. So if you don't have that going, you're in trouble. So here then is we move to maintenance and expansion of the further population information per se. This is a map of the Crescent Creek watershed. This is a distance of about 1,700. It's about 3/4 of a mile, I guess, a little bit less than that. About 1,700 meters distance here, and it along that area is where this population occurs, and this is in 1977. Remember, it was discovered in 1975, at which time there were 41 individuals. And all the way, so by 1977, it had gone down to 26 individuals. And the important thing is that there are 3 ownerships. One is National Forest land, Jefferson National Forest, which at that time had 2. There's one recruited, and then 2 adjacent land ownerships, private land ownerships, where the bulk of the population is. Notice that it's surrounded by, it's hard to tell exactly what's going on, but the white is basically surrounded by agricultural fields and residences, people's residences. And so it's really confined in the area that it's in. So how do you move it, beyond here naturally when you have these human-induced barriers to migration to colonization is the case here. So this population then went down to, by it had gone down to 26 individuals, I said, by 1977. And most of that was due to human factors. This is where the human dimensions come in. Specimens that were taken for research purposes, vandalism to the propagation, landowners, the private landowners, moving plants because they own the plants. That big thing that, coming out of English law, right? That the English law was the organisms that move belong to the state, and those that don't move belong to the landowner. And that's carried over to the present in the Endangered Species Act, such that these landowners could go out there and do anything they want to these federally endangered species. There's not a thing you can do about it. And it's sort of an interesting dichotomy, and it set up some real challenges with people who work with conservation plants versus people who work with conservation animals. So by 1994, we were down to 10 individuals in the population. And most of that was due to natural factors, stress in these environments along the creek beds are very dry most of the year. And being relegated to a understory position by riparian tree species that are growing fast. When I mentioned that, they hit the understory, they're done. And then in 2021, when we re-entered back into this seriously, it had gone down. Actually, the 3 individuals, there were 2, all 3 now, were on federal land, and I show the largest one here of this Betula lenta, which is about almost 12 inches in diameter, DBH, and 70 feet tall. So that's where we were with the natural population at those time periods. And and I might add, and this is a great picture of Crescent Creek here and the environment which these things occur along this creek. And there was protective fencing that was put in here by the Forest Service. This is that same individual that I was just standing next to over there to and the idea was because we were having vandalism on the private, as components of the population, that maybe focusing people on this public thing would decrease some of that pressure on private land. That was the thing at the time. And so we'll talk a little bit more about that. But I also want to mention here in terms of mixed leaf birches in this, that we were talking about in 1979, again, 23 rounding birches, 623 sweet birches, common sweet birch, Betula, and only about 11 yellow birches. So only about 6% of the population is birches. So then, so what we did in that natural population, just based on what we know about how to regenerate birches in general from previous literature that came out from working with Betula and Kansas, is that we made clearings, we created clearings in the area where the natural population was, and we removed the litter life in those areas. And we were very successful in getting regeneration on the private sectors. Public sectors not, because of isolated pollen, not enough pollen, we think, because of some trees on the private sector, and the lack of ingredient and self-compatibility. But these treatments were done to mimic fire, disturbance such as fire, which is typical for these successional species. And the short story is here that by a year later, the population was reduced 30 seedings by the fall of 1980 and to 0 in 1985 due to mainly vandalism. Okay, somebody went in and just took these individuals out. So clearly, the pressure was on to look at alternatives as to how we're going to maintain this population. And so then we turned to these other populations. I could mention we also did selective release competition around individual rounding birches and foresters, but on private land to try to keep them more viable. But one of the private landowners denied us access to the plants, so we could not go there. We just didn't have access to them. It had to do with, I won't name a very well-known arboretum that was accused of stealing plants to propagate them from the landowner. The landowner was not happy about it, and he decided he wouldn't give anybody access. And he didn't do that. And Nature Conservancy purchased the land to expand the area that we can use. So that's when we got this really heightened need to establish, to do mass propagation of seedlings for distribution to the public and private sectors, and to establish these additional, what we would call additional populations at the time, are now called, that we now call progeny populations. And thinking about this in sort of in the sense of assisted colonization or assisted migration, right? Which is a proper term to use where human landscape barriers exist and they can't move because you create this situation. So, and so we did that. And so this is the Crescent Creek watershed. And within that, over about a 10 square mile area, we selected 20 areas to, in fact, establish these progeny populations. And all of them were within that. And this was intended that they would remain within that watershed, not move outside the watershed. And that they would all be on public land, of course. And so they're all on Forest Service land in the Jefferson National Forest. And we put them over a wide range of conditions where sweet birch, the common sweet birch, was known to occur, and using that as a decision point. And I should say several of them formerly on American chestnut, which will come into the story later because we now have American Chestnut Foundation is now working with us on this project. And so If you then look up close then at one of these 20 populations, this is the layout. There are 96 individuals that are in each one of these populations, and they're in rows of 16, rows of columns of 16, and 6 of them, and they're 3.3 meters apart. And that was chosen so that when these were put out as 2 to 3 year old seedlings from 1984 to 1987. And the thinking was that when at maturity, that spacing, we would get canopy closure and exclude a lot of other things, including birch regeneration, by the way, in the end of the story. And yeah, so we did that, and so we put out there 1,960 seedlings, which we were open pollinated seedlings from other trees, you know, in the original population. Vegetable ubers, 6 of other trees of vegetable uber, and 4 of other trees of vegetable lenta we put out. And I just have a note here, 74% of the transplants then were from ground leaf first mother trees, 76% had ground leaves vegetable. And so we, over the years, then mortality, and I say here is to a lesser extent, growth rates, diameters, and heights, and catkin production has been recorded annually, was recorded annually through 1993, and somewhat less frequently through 2003. Most of them in these additional populations, most of the mortality is due to natural causes. Ice damage, deer rubber, animal rubbing, competition, some competition for space, and some vandalism. 23 trees were taken from 6 of these 20 populations over a 4-year period of time. And I'll fast forward to say that we got into mass propagation and made individuals then available to the general public and the local community. The vandalism went away because we no longer had something that was unique. You could just ask us for it and we could get it, right? And so that was really important information as it related to vandalism. I will also say that in terms of Virginia round-leaf birch, vegetable sweet birch, you can see here that in terms of their mortality rates, survival rates, their growth rates, and so forth, there was no significant difference between them. So there's no sort of depression, so far as we can tell, in the round-leaf birch population relative to this very common widespread sweet birch. And then we, in conjunction with maintaining these things, though, we use various methods to maximize survival in these 20 populations, including clearing of competing vegetation, fencing around individual and targeted application herbicides. And this is always a really interesting thing when you're doing this restoration type work or recovery type work. Do you intervene with nature to a degree to increase the probability of success? If we did nothing in these populations, we would get some recruitment, but it would be far reduced over we intervene and do certain things to remove the competition and to increase the probability of individuals surviving. And at this point in time, over half of those individuals survive across those populations. And if it weren't for one area, we established in a pasture that has only 2 of 96 individuals remaining top, the number of survival goes up to around 70%, which is really quite good over 4 decades. And so here we are in 2023, and you're looking at one of these stands right now with Bob Ford in the understory, and I say 75% of the individuals are in the round-leaf birch. The largest individuals now, all of the mortality since 2021 that we've gotten back in here occurred in suppressed trees and animals. Nothing in the oversight has. Whatever reason, they get into industry, gone. And now the largest individuals around 11 inches in diameter and about 80 feet in length in these populations. But no natural regeneration, for at least 3 of these things probably started falling around age 10 or something like that. no natural regeneration since then, not only in the understory of these populations, but bordering these populations, which are forests, right? And as I say, are likely due mostly to a lack of regeneration issues. So, bring the water. This is challenging in itself. Okay. So we implemented then the first of these regeneration treatments in 2023. So think about that. That's 4 decades after we had established these populations. We had to get equal approval was required on federal land clear to do that, and we did. And there's a little interesting little vignette there from the standpoint that the requirements that come out of that, and I'm fast forwarding here for a minute, is that we could not disturb areas within 15 feet of the stream banks. Well, that 15 feet of the stream banks are the very best regeneration niches of this species. And so we're talking about a conflict between 2 conservation values, right? Between maintaining these aquatic ecosystems versus an endangered species, right? And so it's an interesting thing. I tried to talk about this and I was successful. I'm not done with this yet because they're really small areas that we're disturbing. So we do counts of male and female catkins and site quality used to determine which of these populations we could treat in any given year. Okay, and I'm going to show you that they're not all equally combed in any given year. And for the preference is given to producing to those populations in a given year that were producing abundant male catkins flowers in the spring and then abundant female catkins in the fall. And you need both because if you had a bunch of female catkins in the fall with samaras on them, but there's been no pollen, then nothing's going to happen, right? And so we have to continue to monitor that to decide, okay, we did that in 23, and that led to our choosing 5 of the populations that we were going to treat that had sufficient male and female reproductive structures to treat, but we didn't have enough money, and so we just treated one population of 3, which is estimated in that given year, that population was putting out over 5 million seeds. 5 million seeds, right? And so here are maps of scaling for the number of male catkins among those 20 populations. So obviously the bigger the circle, the greater the number of male catkins. And then over here is the number of female catkins. Now these 2 are scaled differently, so you can only compare the size of the circles within one or the other, not across them. But you can see that clearly there are differences among those in terms of who's producing lots of male catkins and who is not. And it switches from year to year. It tends to be somewhat correlated with the subdivision of the watershed, or certain segments of the watershed are producing and others aren't. We do not have climate monitoring in all 20 of these. I mean, if you had a lot of money, you would be monitoring climate and other things in each one of these to really tease out what's going on here in terms of why some and not the others. And so here you're looking at, here's what we, in the fall then, in population 3, we've removed all of the woody plant material and the litter layer, and we're looking towards the individuals in population 3, the 20 of the adults. Here is that 20 by 50 meter area that I showed the map. I should have mentioned the 20 by 50 meters. And then around that, then we created these treatments, started treatments for each 20 by 20 meters in size, and 400 square meters for 1/10 of an acre, into acres, and trying to mimic large gaps. Okay, and this is one of those gaps. Actually, you're looking at one on the side here too. The second one is back there. And so But we had to, you see fewer individuals here because of the stream. The stream is not on the scale, but if it were, you would see that there was a stream coming down there. And so we had to honor this buffer beside the stream. And so I had to do within 15 feet of the stream, right, the aquatic ecosystems. Now, another bottleneck in this is that the treatments had to be implemented between after November 15th because of bat hibernation issues, but before the mainstay of dispersal, which starts mainly in mid-December and goes through mid-February. So you've got this window of opportunity. So you can imagine if you're going to do 5 of these a year, this is really a lot of work to get this stuff out of here. Believe me, trust me. So you got this bottleneck that you're dealing with in terms of time to do these treatments. And these, by the way, these little yellow or oranges, little areas here, in which they're anywhere about 30, usually 30 or 36, are 1 square meter sampling areas. We use microplots along these transects to record the regeneration in those areas. And so here we are again, 1 year, that was in 2023. So now we're looking at the fall of 2024, I mean, the spring of 24. No, I'm sorry, the fall of 24, the end of the first growing season, right after we did the treatment stuff. And here's a really nice photo of a round leaf birch, that's the uber seedling next to a sweet birch. And then here in this 1 square meter frame, which is not 1 square meter because I changed the thing and I stretched it out, but it was, trust me, it was a square meter. But I put in, and so the orange, all of the green flags here are Betulentas, and the orange flag is a Betula. It's hard to see with the scale. But the main reason I put this in here is because of the rockiness, these rock fragments. And it's really, and which you don't see unless you take the litter off, right? You don't see that. But once you do that, you realize that there are a whole bunch of, a large number of different regeneration niches on a micro scale. And it's not all the same. You're looking at the litter over there, it all looks the same. You take the litter off and you see a lot of variability. And these things, they really do really extremely well when they fall in the cracks between these rock crevices, and it creates a micro pocket, I think, of moisture and temperature, cool, convectional lowering of temperatures that provides a really great niche for these things to be successful. And that will show up in our statistics of where, when we analyzed, we measured like 11 different variables in terms of substrate to see what the regeneration was associated with. And one of them turned out to be this percent rock cover. Positively speaking, the higher the percent rock cover fragment, the higher the number. So we did that, and that resulted in, our work resulted in 2.1 birch seedlings per meter squared, and 15% of those being positively identified as round-leaf birches. And these things are really small, so that they can get, it can be really hard to tell. So we were conservative. But that 15% or that number, that density is 0.3, approximates the percentage of adult Virginia roundleaf birch in the population, in the parent population there. Okay, that was contributing the seeds to that. And that number was recruitment there is more than twice that of the original population. So, I think quite successful. It equates to 720 Virginia roundleaf birches for the 6 treatments as a whole, and that's more than adequate to support 270. These things get to maturity. If you were going to completely occupy the site, it would be 270 individuals. So there's, that's true, but they're not uniformly. Here's all the birches collectively, the roundleaf birches and the sweet birches, in terms of their numbers, and then over here are just the roundleaf birches. But you can see that they're not equally distributed by the project area. So it gets in, it gets to the question of whether or not you might want to consider assisted migration on a micro scale, right? Should we move some of these individuals so that these places, these sites can be completely occupied or not? We haven't moved any individuals, but it's assisted migration on a micro scale might certainly be a possibility. You got some things here that are rather interesting, and that is that normally you would expect, and you see this in three of them, that you get this exponential decay thing where most of the seeds are falling close to the parent trees, right? And so you're going to get more regeneration, you should go away from it less. And that's true here, except in it down here, you see just the opposite of that. But it turns out that the main vector of wind is in that direction to the southwest, so it's pushing those seeds out. We had done earlier studies to study how far seeds were dispersed in the direction in another area, and so we had a pretty good idea of what was going on. And so what we did, and I mentioned this already within here, we also set out seed traps in those areas. It was actually done by the Morton Arboretum, and here are the results of that seed trap data that we did. And there's a pretty good correlation in this, except for this population down here. Remember that a high concentration down there, but otherwise, and again, it tends to be that negative exponential decay away from the source of the seeds. Here, we measured all of these different variables on the substrate, the amount of live foliage, leaf litter, soil, bare soil, rock. I talked about water, raw woody debris, decomposed woody debris, stumps and live stems and roots. And then here's the density of vegetable and vegetable lentil plants. The only significant correlations that we got was a positive correlation with the amount of bare rock and a negative correlation with litter. The less litter, the higher the densities, right? So it behaved as we would have predicted, and that's successful. So we cleared competing woody vegetation, just mostly caucus stuff, real fast growing from around those individuals mid-November, and then fencing. We didn't and the idea of the fencing was delayed until the 2025 growing season because the individuals were so small. So we just waited, and you're going to see that we've done this in other places in the past. And the cheapest and the most effective is to take poultry wire, chicken wire, 60 cylinders, 2 meters in diameter with 3 rebars in each one to create this tube around each individual. Otherwise, the deer herbivory can be really extreme. We haven't experienced that. Yeah. So we did no regeneration treatments due to a catkins in 2024. We're going to do it the next year. Remember, we did this in 23. We're going to do it in 24. Well, look here. Here are the male catkins. Over all of those 20 populations, there was not a single tree that produced a single catkin. Not a single one. And so we knew right then, didn't matter. And then over here in the females, there were only 1 to 2 individuals that had catkins and female catkins in 5 of the population that really loved it. And of course, why would you produce female catkins, right, if you weren't having males to pollinate these things? And so, okay, you get funding and you say you're going to treat 3 or 4 populations in each 4 successive years. Well, no, you aren't, right? You're just not going to do it. And so then you go back to the funding agency. Argue that, no, we can't do that because it's just not going to work. Do it, but it's not going to work. So, and so here then, much higher production of flowers and fruits of all progeny populations compared in 2025 compared to 2024. And when we measured the seedlings again, seedling density of seedlings in progeny populations compared to 2024. And here we found a positive correlation of vegetable seed density recovered by vegetation, providing some protection currently. If they're on bare, really exposed areas, they're really vulnerable. And so some percentage of vegetation competition is good. The largest individuals, this is at the end of the second growing season now, is about 3 feet in height. You can see this individual here in the second year. And those are the individuals that were caged. And here you see, this is one of those 20 by 20 meter microplots. And each one of those green flags is a sweet birch, and each one of the red flags is a rounding birch. So it's telling them what the density is. And what we did is we went in and we took out, we started with all of the vegetable overs, and we removed the flags, the green flags and the lentas that were within 1.5 meters. The overs took all of those out, and then just fenced around the flags. We didn't take the fence out. Then caged around all of the vegetable individuals, of which there were about over 210 individuals to be caged, which is a lot of work. And we had a lot of things to do that. And so that was at the end of this 2025 process. Okay, I got about 5 minutes on this. Okay, so I got that. That really is the most important stuff there. This, I already told you, continued searches for the in the natural population where over 3 county area produced no individuals. And then the other area here is determination of systematic relationships with other bird species. And here, just to give you an idea of what this looks like, this is the leaves, foliage, silhouettes of yellow birch, Betula alligator. It's only 11 of those. This is the common sweet birch, very fine serrations on the margins, and then this is brown birch. And down here are those bracts that I talked about, the catkins, the 3 low bracts, like turkey peat here. This is yellow birch, sweet birch, and roundleaf birch. And statistically, you cannot separate between roundleaf birch and sweet birch. And so the only differences are in the foliage. And as you know, most taxonomic classifications depend on reproductive structures. And so that's the controversy over the level of this classified as. And so, and as I mentioned before, it's been classified as a species, subspecies, variety, and form level. And we produced, we did a morphological analysis on 10,000 progeny of brownleaf birch and sweet birch from 10 open-pollinated trees. Over 99% of them exhibit at least typical either of length or uber. No intermediates moved. Okay. And you're going to see, I'm going to skip through here. This led us to the hypothesis. This brownleaf shade is a case of simple Indian inheritance, resulting from the single recessive or maybe a few more genes in their population as the birch population. And but I just mentioned here that a lot of interest in the role of single gene mutations in the higher evolution of their plants. So and then of the controlled crosses that we did that we needed done more, but the ones that we did support that. The ooze or ubers, the rounding birches and the lentas. This is the female parent and the male parent. And you can see here that if you cross this 782 uber with this upper uber, there was all of them, and there could be some contamination of rounding individuals. But then if you look at that 782 and cross it with these 2 lentas, you get 50% of each round and ovate individuals, suggesting that these 2 birches here are heterozygous for that trait, whereas that first individual is a big A thing, right? Versus big A little a. And we're now doing, because of the time, we're now starting into the genomics on this stuff and to really get into this in terms of this relationship between the 2 of them. And ultimately, there's a lot of interest among population geneticists in understanding specific genes that control specific characters. So we want to get to a level where we can identify the individuals. It turns out that the canopies. I mean, there's differences in the branching patterns, much finer in Uber than in Lenta, but we haven't quantified that. It whether it equates to 5% or something like that. So we pointed to the need for a clonal seedling seed orchard, and we established that the place where we produced the seedlings to begin with for the 20 additional populations. It was at Virginia Tech at the research station. It was fine, everything was fine, and then the funding dried up and it went away. And it never got to the point where we could make the control crosses, because for this, in order to identify those specific genes that controlling these attributes, you need at least F1 individuals. You need to get, the first generation progeny, and in some cases you need F2s. So you have to do the crosses, produce those individuals, and then get them up to flowering and cross them again. And these things normally will take 10 years before they begin to flower, which is why we're also experimenting with through the American Chestnut Foundation, because they do a huge amount of control process to work on this issue of precocious flower. So can we accelerate their flowering? So rather than 10 years or 15 years, can we get these things to flower in 2 or 3 years and really reduce that amount of time? And this retention of existing germplasm through cultivation. Was a lot of work that was done on this, both starting with grafting, with vegetative propagation, but of course, we mainly wanted sexual propagation because of the genetic diversity of that, right? And so some of that was done, but we germinated again, several thousand seedlings, and we ended up then in over 2,000 seedlings from controlled crosses experiments that we did. And we distributed these to various places, and 3 plants per recipient, and almost an incubation was provided with them. And as a result of that, we distributed more than 800 individuals between 1989 and 1994 to more than 200 entities or locations, including land people who were in the local community. And that's when this vandalism went away. You didn't have anything that you need all this stuff. And then we had a refugia planting at the Reynolds Research Station, and 800 and 980 individuals is now down to 72 without any maintenance. And then educational programs, just finally would say that we made a decision was made to allow the public to access, as I already mentioned this, on these trees that are on public land, to take pressure away from the private land. We don't know whether that worked or not. The distributing of additional individuals had more of an impact in this case. And so after a while, we just removed that fencing to just get away from showing anybody that we had anything out there because of the vandalism. So where do we go next at the end here? And so in the original population, this is what we're proposing to do now. Is the original population to do continued monitoring of those populations, to release competition around individuals, and to do regeneration treatments in the original natural population. And then in the progeny populations, the same sort of thing. We need to do regeneration in 19 of the 20 population. We don't need to. The recovery plan call for getting regeneration in 10 of those populations. We just put in 20 as a safety valve, and then there's quantification of branching pattern. And then development of a database management study. Another really interesting story here, and nobody told me about this at the time. Nobody was talking about this, but most of you don't go back to the old floppy disks. I mean, the really old floppy disks. And so we had established this data management system and had put all of this information into that data management system that anybody could access easily. And that's a really important concept. It was all in there. And nobody told me at the time that the lifespan on these things is about 8 years. And so we lost all that data. We have most of it in hard copy. It's a massive amount of data that needs to be put back in. But now you could go to the cloud, right, or whatever, but you couldn't there. And but I would still say, it's not bad having stuff in hard copy even now. And so we're going to do that, and then and Suresh is taking the lead on that. And then we're going to establish, we're going to go back around now and establish what we're now calling a conservation orchard to make controlled crosses between Virginia Valley birch and sweet birch. Then that is going to happen in the American Chestnut Foundation research farms, which are about 30 miles from our site. And they do lots of that work. And they've got, American chestnut, of course, is one of the most charismatic species in North America, right? And they know what they're doing. And so we're going to work with them. And they also were experimenting with approaches. And then finally, some successes here I'll point out that in the literature. Now this may have changed, but at the time, Betula uber, with only one of two species, quote unquote, that can be considered as having been reclassified from endangered to threatened primarily because of the Endangered Species Act. Others had been, but because of funds from other places, not because of the Endangered Species Act per se. And then secondly, another quote here, that it can be considered perhaps the most successful case of assisted colonization of a plant species under the ESA. And that was the establishment of these 20 populations that we get. And I can tell you that the Fish and Wildlife Service is looking for success stories. From a political standpoint, they need success stories. Okay, I talked about some of the minimum funding levels here and the long-lived plants or long-term plant strategies and actions to sustain them. And that's really it. I get here along the way who's a bioinformatics guy and has worked a lot with the genomics of Betula, offered up. You could ask the question, well, why? Especially if it may not be a good species, right? And other than the fact that it's under the Endangered Species Covered Act, it's been threatened, it's been moved, threatened and endangered at the federal and state levels respectively. And he offers up these from a sort of a genetics race standpoint in terms of genetic diversity and ecological stability and evolutionary potential and hidden genetic and molecular value as reasons as to why we might want to be interested and concerned about the species. Okay, well, there you have it. References here. And here I am, and that's 50 years of